Hi, everyone. My name is Abbey Wilson, and I am a postdoctoral fellow at the University of Saskatchewan and FRI Research. And today we will be discussing our study on the development and validation of protein biomarkers of health in grizzly bears. So grizzly bears, or also known as brown bears, are distributed across North America and Asia, with extant or resident populations shown in orange here on the map. Globally, grizzly bears are classified as least concern because their population is considered to be stable, despite the areas there on the map shown in red, which are where grizzly bears have gone extinct. Our study population resides in Alberta, Canada, which is broken up into 7 bear management areas, or BMAs, which are distinguished by the major highways that run through the province. You can see that there are extremely high levels of human disturbance and landscape change, such as roads, rail lines, cut blocks, coal mines, and oil and gas well sites. And these have all been increasing over the years due to the demand for natural resource extraction in this province. This ultimately will lead to habitat loss and destruction for our grizzly bear populations. And so this species was listed as threatened in Alberta, Canada in 2010. And this led to significant work on the conservation and management strategies to recover this population. One of these conservation and management strategies was to assess the health of individual grizzly bears. So in the last 10 years or so, our team has measured biomarkers in hair and skin using various techniques. We did start to look at protein biomarkers in particular, but when we first started, our technique would only allow us to measure the relative concentration of proteins. So we couldn't really have a quantitative method. So we wanted to apply the use of these new contemporary molecular tools, so things like targeted proteomics, which is often used in human medicine, but in our case, we wanted to apply it to wildlife. Using this technique, we could get a more accurate measurement of the protein biomarkers, and we could look at multiple biomarkers at once in fairly complex samples. There have been a few attempts to identify protein biomarkers in animals. These include those indicative of pregnancy in domestic dogs, cheetahs, wild canid species, black bears, and polar bears. And because skin is so highly involved in day-to-day maintenance, and it can also be potentially collected non-invasively through remote biopsy darting, we aim to first identify proteins that are detectable in the skin of grizzly bears, and second, to determine the expression of proteins related to energetics, reproduction, and stress in skin. Now, given that proteins are extremely large molecules and they respond to stimuli, and that they've been used as biomarkers in human medicine before, we hypothesized that proteins could be measured using liquid chromatography, tandem mass spectrometry, multiple reaction monitoring, or LC-MS-MS-MRM, and the expression of these proteins would be influenced by biological variables, so things like sex, age, and sample location on the body, as well as environmental variables, so geographic area, season, and sample year. So we look for biomarkers of health in skin because skin has been shown to have this unique process for responding to stress and other stimuli. And we think that it's equivalent to the relationship with the brain and adrenal glands. Skin cells will produce elements of the HPA and the HPT axis. So HPA being the hypothalamic pituitary adrenal, and HPT, the hypothalamic pituitary thyroid axis, both of which will regulate metabolism and the stress response. Skin cells will also produce elements of the CRH-POMC system, steroids, and biogenic amines. So if we zoom out a bit, you can see that there are nerve endings in the epidermis, adnexal structures, so things like hair follicles, sebaceous and apocrine glands, and stimuli by neurons, which all contribute to the exchange of neuroendocrine mediators between skin and other organs. And this helps to regulate homeostasis and the response to signals. So during our kind of ongoing and routine capture efforts, we had collected about 140 skin samples over 7 years from either an earplug or a biopsy from the outer thigh of our captured bears. We initially identified over 700 proteins in grizzly bear skin, and these were mostly related to skin structure, glucose metabolism, and the inhibition of proteases, which are just enzymes that break down other proteins. And since then, we've developed and validated our LC-MSMS-MRM assay for 19 specific target proteins related to metabolism, reproduction, and stress. So in order to quantitate the expression of proteins in skin, we first needed to identify our target proteins and their corresponding unique peptides. Next, we digested the proteins with trypsin to separate the distinct peptide so that each protein was represented by 3 unique peptides. We then created a standard curve for each peptide using isotope labeled peptides. And fourth, we analyzed the samples using a triple quad LC-MS at the University of Victoria Proteomics Center. And the way this works is that the compounds are initially separated in the first quadrupole, and this results in a precursor ion. And then the precursor ion is further fragmented in the second quadrupole, which is also known as the collision cell. And lastly, the fragments are further separated in the third quadrupole from the collision energy in the mass analyzer, and ultimately are detected by the mass spectrometer in the end there. So the transition, which is the precursor ion over the product ion, is unique to each one of these peptides. And the amount of the production ion will tell you the amount of corresponding protein expression. And lastly, we quantified the concentration of unique peptides in our unknown samples using our standard curve. So out of the 700 proteins that we initially identified, we narrowed our list down to the 19 specific target proteins, and we thought that these would be good biomarkers of health. First, because they had these established relationships with physiological processes from the scientific literature. And second, they had potential to answer specific management questions that we were interested in. So we identified proteins related to metabolism and energetics, which regulate glucose, transport nutrients, and clear cellular debris. And these can serve as biomarkers for metabolic disease, malnutrition, and inflammation. We chose 7 proteins related to reproduction, which are involved in transporting nutrients for pregnancy, clearing pathogens, and initiating parturition. And these can be used as biomarkers of pregnancy, female fertility, and pregnancy disorders. And lastly, we chose proteins related to stress that are involved in transporting stress hormones, maintaining protein function, and reducing inflammation, which can be indicative of impaired immune response, inflammation, and infectious disease. Now, of course, all of these can have a negative impact on population health, cub production, and survival. So now that we had our targets measured, we wanted to know if these would be good biomarkers of health. So we expected them to change relative to certain variables rather than just being highly expressed all the time. We used a generalized linear mixed model analysis for each target protein to determine the influence of biological variables on expression. So we orient ourselves to the graph here. We have protein functional category on the x-axis and the number of proteins that were influenced by these different variables on the y-axis. And the different variables are just represented by the different colored bars here. We found that sample location on the body, so ear versus thigh, influence the majority of proteins. So we just need to be consistent with our sample collection body site in the future. We also found that sample year was strongly influenced by the expression of all proteins. And we think that this might further suggest that there's changes in the environment from year to year that are influencing physiological function. We also determined that season, based on grizzly bear feeding habits in the spring, summer, and fall, influence the expression of proteins related to metabolism, reproduction, and stress, while geographic area influenced those related to energy, metabolism, and stress only. And not surprisingly, biological attributes, so sex and age class, affected proteins related to reproduction and stress. So for this study, we used LC-MS/MS-MRM to measure proteins related to metabolism, reproduction, and stress in skin. The expression of these biomarkers was influenced by sample location on the body, year, season, and biological attributes. And since this study, we have applied this technique in two different ways. The first was to identify biomarkers of reproductive status, and hoping that these can answer some important management-focused questions. And we found that biomarkers were indicative of pregnancy, lactation, and sexual maturity. And the second was to determine the landscape variables that drive changes in physiological function. And we found that protein expression was related to landscape variables that mostly influence food resources. So things like coal mines, crown closure, percent conifer. And the expression of metabolic and reproductive proteins increased in bears that spent time in areas with more foods. And stress-related proteins increased in bears that spent more time in areas where there were lower food resources. So this study was published in Conservation Physiology in 2020. So if you want some more information, you can check it out there. And with that, I thank you very much for your attention. And I thank all of our funding sources and the village of collaborators and colleagues that it takes to study this grizzly bear population. And feel free to reach out and connect if you have any other questions about this. Thank you.
Hi everyone, my name is Melanie Clapham and I'm a postdoctoral researcher at the University of Victoria. And I'm going to be talking to you today about our project developing face recognition for bears. So a quick project overview to start with. We're a collaborative group of ecologists and software developers. We came together to develop this technique that is robust, open source, and can automatically identify individual bears. And our aim is to achieve over 90% accuracy. We started off with focusing on brown bears, using a wild data set of DSLR images. And we achieved a detection rate, which is the detection of the bear's face in the image, of 0.98 or 98%, and a classification rate, which is the identification, of 84%. And we continue to build upon this level of performance. But why is it important to be able to identify individual bears? Well, if we were to combine BearID with camera trapping, we believe it could be used to help to establish population estimates, to look at movement patterns of bears, to better understand behavior, and also better understand human-wildlife conflicts and coexistence, which also has management implications. And a critical part of automated photo ID is to be able to detect the animal in the image. And this is important for standardization across images. And we use a form of machine learning called deep learning to be able to automatically detect bear faces in photos. And similar studies have used machine learning to detect things like the ear contours of an elephant for individual ID, the flanks of a giraffe, and also the face of a gorilla. And some detection networks use multi-species approaches. And a really good example of this is the mega detector, which detects animals in photos. And researchers can use the mega detector to reduce the amount of time it takes to process camera trap images, and also as part of identification, what's called pipelines. So being able to use these images for identifying individuals or species. One of the benefits of this multi-species approach is that it is able to help speed up the development and application of identification systems across species. So with this in mind, we set out to develop an automated multi-species face detector across all 8 bear species. And we did this using an existing data set of images provided by colleagues at San Diego Zoo Wildlife Alliance for other research, and also 2 rescue centers from Europe to collate a data set of images of bears taken under human care. And here's the number of images that we collected per species and the number of individuals that those images represent. We collected a total data set of 2,192 images of 445 individuals, and we split that data set into 80% of those images for training and 20% for testing the face detector. And here is how our Bear face detector works. So you input an original image, our sub-application Bear face then finds the face, so that's the box around the face, and the facial landmarks in the image, and then sends that information to another sub-application called Bear chip that then extracts that face and facial landmark information and optimizes the face ready for use in a full ID pipeline. And here's some of our results to start with then. So we use two metrics of performance to assess the outputs of the face detector. The first one is AP, or average precision, and the second is mean ND, so mean normalized distance. And these are two metrics that look at the difference between a gold standard data set, as it would be called. So that's manually labeled information of where the bear's face is and where those facial landmarks are. And we have to do that before we do any training or testing so that we have something to compare the outputs of the face detector to. So both of these measures assess how close the gold standard data set is to the predicted data set by the face detector. An average precision concentrates on the whole face, so that's the bounding box you can see in these images, and the mean normalized distance concentrates on the distance between the outer corners of the eyes, the facial landmarks. And with average precision, we're looking for the higher number the better, so as high a number as possible, with a maximum value of 1. And for mean normalized distance, we're looking for the lower values, so with a minimum value of 0. So overall, we had 438 test images. The detector achieved an average precision of 96% and a mean normalized distance of 8%. And as you can see, from across the different species, the species which we had higher test set data, the species that we had a higher sample size for generally achieved better results. How transferable is a face detector that is trained on images of bears under human care to images of bears taken in the wild? Well, to test that, we used an existing test set of brown bear images, which we collected for our previous paper, and we tested it using the multi-species bear face detector. So here you can see the results of our wild brown bear trained and wild brown bear tested face detector from our previous research. And we achieved an average precision of 98% and a mean normalized distance of around 11%. And then when we run those same test images through the multi-species captive trained detector, we achieved an average precision of 93% and a mean normalized distance of around 16%. So as you can see, those numbers have reduced slightly, which we would expect, but they're actually quite a bit higher than what we originally anticipated. And we think that that could be because of additional variation introduced by different types of enclosures of bears under human care across the different species, and also additional variation brought in by the inclusion of multi-species, so taking into account differences in the facial biometrics of the different species, which could have helped us to achieve a slightly higher precision, average precision and mean normalized distance than we would have expected. And then lastly, then we wanted to test the multi-species bear face detector on a full pipeline of individual identification to see how well it performed. And so we decided to do this for Andean bears. Now, Andean bears do have unique facial markings that are used in manual identification, but these facial markings can change slightly over time, as you can see in these two sets of images of two individuals. And researchers have also expressed to us the need for a more standardized approach to individual ID, and that goes across for all bear species as well. So here is the full BearID pipeline. The blue boxes on top are the newly trained networks through this current research. So the first one shows that we had a new network trained for bear face, and that is the multi-species face detector. So the whole process is to input an image, and then that is fed through the multi-species face detector. Bear chip then extracts that face and sends that extracted face to another sub-application called BearID, which is our newly trained Andean bear face encoder, which creates a vector representation of the face based on prior learning during training, and then sends that vector representation through to the last sub-application called BearSVM, which then classifies which individual or predicts which individual it thinks is in the image and outputs an identification of that bear. And so I should say also at this point that we work on a closed set approach at the moment, whereby all the IDs that are outputted by the system are bears that have been seen previously during training. We are working on what's called an open set approach, whereby the system could identify new bears that haven't been previously seen. But that's a much more complex method to do that. So we're still working on that at the moment. So some of our initial results then, we used 488 training images and 121 test images for 32 individual Andean bears. And we acknowledge that this is quite a low sample size for this kind of technique. But we really are just using it at the moment to prototype the inclusion of the multi-species face detector in a full BearID pipeline. And we hope to be able to build upon this sample size in the future. And so Andean BearID was 91% accurate at being able to identify whether two images of an Andean bear are of the same bear or of different bears, so whether they are matching or non-matching, and 86% accurate at correctly identifying the individual in the image. And here's a visual demonstration of how Andean BearID works. And the green boxes indicate a correct identification, and the red boxes indicate incorrect. And as you can see, when the bear is face on, it's pretty good at being able to identify the individual correctly. But once the individual turns its head slightly, we do see some more inaccuracies, and we lack detection when both eyes aren't viewed in the video. So going forward, we want to try and increase the sample size. And if we include some more images of bears in various types of facial poses, that could help to increase the accuracy when bears are in more extreme poses, as we've just seen. So next steps then, we want to look at concentrating on some of the species for now that we have a bigger data set starting to accrue. And at the moment, that's for Andean bears, brown bears, polar bears, and starting to look at sloth bears and American black bears also. But in the future, we hope to extend this to all bear species. And this is what we're aiming for in the end, to be able to integrate BearID into camera trapping so it can be used for research and monitoring of wild bears. And with that, I'd like to thank my co-authors, our partners and supporters on this project, and thank you for listening. And I look forward to taking any questions.
Hi, everyone. Thanks for joining my talk. My name is Ali. I'm going to be presenting part of my master's thesis work, which dealt with understanding bear movement, learning, and memory using an agent-based model approach. I was part of the University of Victoria, the Zubria lab. We collaborated with folks from the Foothills Research Institute in Hinton, Alberta. So as you all probably know, grizzly bears once ranged down to northern Mexico and are now currently only found in northwestern Canada and a few small populations in northwestern United States. And the landscape that the grizzly bears are experiencing is not only already extremely developed, but it's increasingly more developed and more just inhabited by humans. So it's really important for us to understand how grizzly bears are interacting with the environment and how they're going to have to adapt to new conditions in the landscape with more development. The project that I worked on focused on bear management area number 3, which is located in the Yellowhead region of West Central Alberta. It's an area that ranges everywhere from protected areas to highly developed areas, so it offers a wide range of landscapes for grizzly bears to move throughout. There's been a lot of research in this area already, some of which has looked at grizzly bears' reactions to different linear features throughout the season. So for example, here we see that grizzly bears are selecting for certain features in some seasons and then avoiding them in other seasons. And a lot of this can be explained with food resources, but reactions to features are not always very simple. A more recent research from the University of Victoria looked at the difference in scale response to features. So for example, here, the reaction to road density at a habitat level, grizzly bears are decreasing the selection of habitats that have increasing road density. However, at a small scale, we see that bears are actually preferring areas that have increased distance decay to roads and increased edge density. So it is important for us to consider not only seasonal variation and all of these factors, but scale as well. Limitations of the current research, the main one is that the relationship of the positional data to the landscape characteristic is often very static. Movement is often considered a response to something as opposed to a process. Optimization algorithms are often not realistic, one of which is Markov-based algorithms, which ignore learning and memory. So in order to approach a different way so that we could deal with the limitations of current research, my project is focusing on agent-based models, which simulate individuals' decisions. They're dynamic and interactive, so each moving agent can interact with the environment and the environment can change throughout a simulation. This allows us to create a highly explorative simulation that acts as a virtual lab where we can test different scenarios. And the concept of emergence is really important in agent-based models, which is the idea that each individual decision is ultimately going to lead to a population level process. So for example, here in this video, we're simulating bird flocking behavior. And right now birds aren't following any rules. So what we see is highly random movement. On this next video, we ask birds to follow 3 simple rules based on their alignment, cohesion, and separation to other birds. And what we see is that very quickly flocks are forming in the birds. So each individual following very simple rules leads to population level behaviors. Some agent-based model examples in our field include animal migration, animal learning and adaptation, cognition. Work has been done with human-bear conflict management, as well as landscape connectivity, which is the example I show here. This just shows a map of current landscape connectivity for bighorn sheep based on current landscape management. And then panel B shows landscape connectivity where management actions that restore natural fire regimes have been implemented. So it allows us to really test for predictive models as well. The research goal for my project was to understand how bears acquire information from their environment and use it to make informed movement decisions. And for this, the objective was to develop an agent-based model that simulates grizzly bear movement behavior using multi-scale learning and decision-making. This is novel because although there are models that have studied movement, machine learning, memory, and have applied this to conservation, my project brings all of those together in order to present a more holistic approach. And we are able to exploit a huge source of GPS data that was provided to us by the Foothills Research Institute that allows us to corroborate what we find and really make sure that our model is accurate in representing grizzly bear behavior. So for the model, we focused on a small area of the grizzly bear management area number 3 in Yellowhead. It is about 11,000 kilometers squared. It is a dynamic landscape that changes throughout the seasons to represent differences in food availability, and it is multi-scale. So we modeled the landscape at 2 different sizes, one where the cell grid is 1 square kilometer, and then overlapping those cells, we have cells that are 100 square meters. And these cells each have a utility value that represents the value to the bears. And it brings in factors such as food availability, topography, landscape features. And so they represent the utility of the landscape to the bears at 2 different scales. We simulated a single female agent for 30 years and repeated simulations 100 times. And we used an explore versus exploit approach, where at the beginning of the simulation, bears were exploring their surroundings and just gaining information about their surroundings. And then in subsequent years, every movement was more so exploitative, which meant that the bears were using information about the landscape to move, and hence they were following landscape cues. We tested 3 scenarios, the first with random movement, where bears only explored, the second where bears were learning about their environment, so they were actually able to use the utility values to make movement decisions, and a third scenario where bears were using utility values, but they were also remembering previous experiences and able to use that information in subsequent movement choices. At the start of every year, our bears move to a specific starting point, and we reset all of our parameters, and I'll talk about a little bit about those a little later. During the movement, bears were either exploiting, which constituted random movement, and there would exploit, which basically meant that if the patch that they were in at that moment was depleted, so if the utility value was below a certain threshold, the bears would seek a nearby patch with a high utility value and move towards it. And if the current patch was not depleted, they would just exploit during within that patch. After each movement decisions, we would update the utility value of our patches so that if bears were in it still, the utility value was depleted following depletion of resources, or if the bear had moved away from the patch, then resources would start to replenish. We also calculated a measure of success, which was just the cumulative quality of all of the patches that had been used during that time, so that we could measure both seasonally and yearly how efficient our bears were at accessing high quality areas. At the end of the season, we went through a reinforcement step, which basically meant that we were comparing the quality used of cells used during that season compared to the cells used during random movement. And if the cells used during exploitative movement were better, then they were positively reinforced such that the utility of that patch was more so. This just meant the bears were more likely to return to previous areas that they had used if they had found high quality resources there. For our results, we plotted annual measures of success at both global and local scales, so large and small scales, and we evaluated differences between scenarios and changes throughout a simulation. So you're going to see plots like this, which basically show each year the distribution of quality that each bear had. At the top, we have global, so large scale. At the bottom, we have small scale results. So what we can see is that during our random scenarios, the quality access, the measure of success did not change throughout the simulation. It remained pretty constant and it remained pretty low in both scales. However, whenever we incorporated learning, be with or without memory, we saw an increase in the quality the bears were accessing throughout the years. So this meant that as soon as bears are starting to exploit their resources and use landscape cues to choose where to go, they're going to be accessing higher quality habitat. We also compared home range sizes, and what we have here are the 3 different scenarios: random learning and learning with memory, with a yearly annual home range size for the 3 scenarios. And then in the blue lines, I'm showing the mean and standard deviation of radio-collared female home range sizes in this area, which we obtained from the GPS collar data. And what we see is that it's only the learning with memory scenario that simulates home ranges that are equivalent to home ranges sizes from female bears in the area. And this was really important because although learning and learning with memory both resulted in high quality usage, it's only learning with memory that actually results in the emergence of home ranges. And this is just a visual representation of that, with the red polygons being home range sizes for female bears in yellowhead bear management area. And then the black polygons are simulated home ranges. So we can see that they're pretty comparable. So our conclusions were that agent-based modeling is a useful tool for studying complex processes and for simulating grizzly bear decision-making. We saw the emergence of home ranges with our learning with memory scenario, and our results were consistent with previous research. If you do want more details on this, because it is a very complex model, I encourage you to look up our recently published manuscript in ecological modeling. Our next step, work in progress, is to understand the difference between resident and translocated movement. So what we see is that resident bears have very defined home ranges. This just shows resident bear home ranges between 2016 and 2017. And on this figure, see here, we have location points for translocated bears during 2017. We're seeing that translocated bears have higher movement rates and much larger home ranges. And we want to understand what it is about the landscape and the way the bears are moving in it that makes this difference happen. So for this, we want to understand the role of behavior and habitat familiarity in grizzly bear translocation success. And for this, we're going to simulate translocation scenarios and evaluate the role of environmental familiarity and behavior on the success of grizzly bear translocations. So this is work in progress, and we'll be working on this. So stay tuned for the application of the model to translocation bear management. Just some cited work, and I want to give a big thanks to the Surreal Lab, the FRI research team, as well as other folks at the University of Victoria that helped out with this project. Thank you so much for joining. Have a great day.
Hey, welcome everyone. I think everyone is out of the waiting room now. Thank you for joining us for our first question and answer session today. We are going to be talking about the Applied Conservation Solutions section. We have three wonderful speakers with us today. I also want to thank our gold sponsor, CounterAssault, for sponsoring the conference. Before I get started, just a few housekeeping items. If you have a question, feel free to either put it in the chat or you can raise your hand and we can unmute you and you can ask the question. Right now, everyone except the speakers is muted. So you will need to have your hand up if you want to be unmuted to ask a question, which we definitely encourage so we can get some good discussion going. So to start off, we are going to have each of the speakers introduce themselves. So we can go ahead and go around and I guess, Melanie, would you like to start?
Sure. Hi, everyone. My name is Melanie Clapham. I'm a postdoctoral research fellow at the University of Victoria, and I'm calling in today from the traditional territory of the Suk First Nation. And my background is in behavioral ecology of bears, focused on olfactory communication of grizzly bears. And at the moment, I'm working on the BearID project, and that aims to develop automated, non-invasive technologies to help to study bears and other wildlife. And that's what I'm going to be, well, that's what my presentation is on for the conference. And the title of my presentation is BearID Automated Facial Recognition for Individual Identification of Bears.
Great, thank you, Melanie. Ali, would you like to go next?
Sure. Hi, everyone. I am Ali. I am currently a PhD student at SUNY ESF in Syracuse, New York. Just started my project looking at population dynamics of the Great Lakes wolf population, particularly how harvesting and other factors of anthropogenic mortality are affecting population dynamics. Previously, I did my master's at the University of Victoria with Dr. Chris Bone. I was building a simulation model to understand how grizzly bears learn about their environment and use this information to move. And right now we're working on applying my model to understanding translocations in bears, particularly the effects of behavior and familiarity with their habitat in the successive translocations. And I've also worked on a few other projects, vegetation restoration stuff in Yellowstone following the wolf reintroduction, and some lion space use modeling in Africa. And yeah, just really excited to be here, and thanks everyone for joining.
Awesome, thank you,
and the title of my talk. Sorry, yeah, my talk is titled Understanding Bear Movement, Learning and Memory, an Agent-Based Model Approach. Thanks.
Awesome, thank you. Abby, would you like to go next?
Sure. Hey everyone, my name is Abbey Wilson and I'm a postdoctoral fellow with FRI Research and University of Saskatchewan. I'm based out of Hinton, Alberta, and work with FRI there on the health component of our grizzly bear research. So for the last 3 years or so, we've been trying to develop novel techniques to try and better understand and monitor the health and physiological responses of our grizzly bears, mostly in response to the human use in industry that's on the landscape in our study area. So I went to Mississippi State with Lindsey and graduated from there in 2017. Yeah, and then made it up to Canada and have been postdocing there. So thanks for having me. I'm excited to chat about this stuff.
Great, thank you, everyone. So now I guess we will open it up to questions. So if anyone has anything they'd like to put in the chat, go ahead. Or if you're more comfortable raising your hand, we would love to hear from you. So we'll open it up now. Not seeing any questions. Come on, guys. Oh, let's see. It looks like Mariella has a question. Let's see. Let me unmute her here. Sorry, trying to figure out the technology. Finding on the list. Kate, do you know if there's a way to get the hand raises to the top?
Just unmuted her.
Okay, great, thank you.
There's a search button at the top so you can search the name.
Great, thank you.
Should I ask?
Go ahead, Marielle.
Hello. Hi, everyone. Hi, Lindsey. Hi, Abby. How, Ali? So I really liked your talks, really enjoy them. I think I'll just start with one for now, for Abby. I was wondering how easy it would be to basically take that technique and apply it to other bears, obviously, because I work with American black bears. So I was wondering if it was like very species specific, or if you think it would be reasonable, or it wouldn't take too much to change that technique into black bears or other species, right?
Well, that's a great question. So the biomarkers that we look at, those protein molecules, should be similar across all mammalian species. So as far as like what those physiological biomarkers would tell you should be something similar. As far as sample collection goes, I've always really wanted to explore the remote biopsy darting aspect, because that's really where we started, was we were trying to find these non-invasive approaches. And right now, for this preliminary study, we were just developing the technique. All of those skin biopsies came from captured bears. So it'd be interesting to do, like if you were doing den checks or something and you could get a skin biopsy or something like that without actually having to capture an animal, that would be really interesting. But yeah, I think it would all be applicable to any mammal. When you start getting in other taxa, you might have to change what biomarkers you're looking at.
Just and what's the smallest sample you can get that could work, and also how old how can you store them for a long time, and would it still work?
Yeah, so the smallest we've done is 4 millimeters, which was enough, but that was like right at the minimum. Like we need really the 6 millimeter biopsy punch is the best. We did extract some protein concentration from 4 millimeter punches, but it just wasn't as good and wasn't enough protein really to put on the instrument. And then as far as how long it would last, the tricky thing with protein work is that it has to be cryopreserved. So you have to get it on -80 as soon as possible. But once it's on -80, for us, I mean, I had it on dry ice. Like I took a little chunk of dry ice with me to the capture site. So I put it directly in my cooler, and then it was on dry ice for maybe a week or two until I could get it back to our -80 at home. So, I'd say within definitely at below 30 minutes, we haven't done any of that testing. I haven't tested any of that stuff. Yeah. But as far as the literature says, that as long as you get it on there and you keep it on there, it's pretty stable.
Okay. Cool. Thank you.
Yeah, thanks. Oh, nice to hear from you.
Yeah.
Great. It looks like our next question is from Carlos. I'll unmute you now.
Hello. My question is to Melanie, and it's I wonder if you have considered adding the ears of the bears as reference points, and if that could be of any help to identify them in when they are on the side, yeah, on the profile pictures.
Hi, Carlos. Thank you for your question. Yeah, actually, when we first started the project, we did a bit of a comparison, working with a different collaborative group, and we actually looked at ears. And we actually had a lower level of accuracy by taking ears into account. We had a higher level of accuracy just concentrating on the eyes for those facial landmarks. So we decided just to go with eyes going forward. And I suspect maybe it's just because of how variable the ears can be, you know, because bears have, you know, these different ear postures as part of their communication. So we found that there was just too much variation from a front kind of facial profile. We haven't tried it using ears from a side profile point of view. We may look at that down the road. We may look at some side profiles. We're interested in exploring like whole body side profiles as well. But yeah, for the face, we found the ears were just a bit too variable. So it was reducing the level of accuracy that we were getting.
Okay, thank you.
Thank you.
All right, it looks like our next question came in the chat, and I think you might have already answered part of it, but it's directed at you, Abby. Do you know if dried skin, salted skin, aged skin, basically the condition of skin samples will affect the analysis, and how big of a sample would you need?
So yeah, we talked about the sample, how big of a sample, really 4 millimeters is the minimum. And then as far as your first question, it's really interesting, and I'd love to do like more preliminary work on it, because dried skin is something that I'd love to try and figure out if it would be a good matrix for doing protein work. Theoretically, it shouldn't work because your proteins would degrade if it wasn't put on -80 right away. But we did do a little preliminary LC-mass spec with some dried skin, and we're actually able to detect the majority of proteins that were in our target panel. So I got really excited about that, but that was just kind of like me messing around, nothing serious. So I haven't really tested it for sure. So I don't really know the answer to that, other than in the literature and historically, it needs to be frozen upon collection.
Great. Thank you. Our next question is from Kristen Phillips. I'll get you unmuted here.
Hi, this question is for Abby. In your presentation, you mentioned how location on the body can really impact variation in your results and the levels of the different markers. So you say you're interested in looking into the darting, the non-invasive method. So how would you account for the variation in body location? Because when darting, it's often you're getting whatever shot you can at the time. And did you notice any trends in or any apparent relationships in particular to like the location on the body, patterns, I should say? Did you notice any patterns in how that impacted? Because you just in your presentation mentioned it impacted, but did not share any potential patterns of how it did so.
Well, that's a great question. Yeah, so we had samples from earplugs and from outside of the thigh. So back a few years back, when they were still putting ear tags in our bears, they would just collect that earplug skin sample, basically that chunk that they got. And then at some point they switched to not doing that and then just taking them from the thigh. So I only had samples from the ear or the thigh. I didn't have them from across the body. So I can't speak to like shoulder, thigh, rump, that type of thing. But yeah, I think for all the proteins, the expression was higher in samples that were collected from the thigh versus the ear. Which biologically makes sense. There's more blood flow, there's more muscle, there's more mass. Like, I wasn't surprised that that was the case. And I'm hoping that if we do try and do some biopsy darting in the future, we would have to test, of course, like rump, thigh, that whole area. I don't know if you would get the shoulder, but I don't think you would get an ear I've never done it, so I don't know. But I'm hoping that if you're getting in like that general thigh rump area, then there wouldn't be as much variation, but it's something that we'd have to test for sure.
Great. Our next question is from Sanzar.
Hi, do you hear me?
Yep.
Hi, all. Yes, my question for Abbey. For your analysis, it's possible take skin from only immobilized animals, or it's possible take skin from prey, from animals, hunting animals.
Yeah, you could definitely opportunistically collect skin samples from hunted animals if you collected the sample, you know, upon harvest, and then if that sample was immediately put on dry ice or to transfer to a -80, yeah, you could definitely do that.
Okay, thanks.
Because that skin, and again, we're not sure the time period that it exactly reflects, but it would reflect, you know, the time and the movements and the behaviors that they were doing previously. So, yeah.
Okay, thanks.
Great. Our next question is in the chat, and it's also directed at you, Abbey. The question is, have you found any interconnections of some of the proteins from the energetic group with the stress-related ones? Or has this not been explored?
Hi, Aga. Thanks for your question. Have I found any interconnections between them? I'm not sure. Interconnections, like if they're both, like if metabolomic proteins and stress proteins were high at the same time. Is it okay to unmute her just so she can, or if you want to talk, Aga?
Yeah, definitely.
If you're asking if we were looking for any correlations between them, in a later study when we applied this technique to looking at how landscape variables influence protein expression, there were some proteins that were correlated, which makes sense, if that answers your question, but I'm not sure.
Sorry for not being very clear. What I mean is basically what you just explained a little bit. I was just wondering that, like, for example, cortisol binding protein would be expressed in times of stress, but then some of those energetic proteins would be connected because cortisol is affecting metabolism quite quickly as well. So I was just wondering how to, because the models, they take into account some other features, but then what it would be in between.
Yeah, exactly. Yeah, I would expect some of them to be correlated, and we explored that a bit more once we actually applied that to those landscape variables and stuff. But you know, and we can chat about it some more later, and I can share that other paper with you.
Okay, sorry if I haven't noticed something then in your presentation or somewhere. Then sorry for the question.
Oh no.
Anyway.
Not at all.
Thank you. Thank you.
Yeah, thank you.
Great. Our next question is from Sarah.
Hi, good morning or good afternoon, everyone. Hi, Mel. I love your BearID work, Mel, but my question is actually for Ali. Okay. Ali, I have been really curious about bear learning and memory and habitat use and how these things all intersect for a long time. So I loved your talk, and even though I didn't fully understand the complexity of the model, because that is not my strength, the fact that you created it and made it is amazing. So thanks for that.
Thank you.
I think one of the things that I've learned in my work, and I think a lot of other people are starting to highlight this, is that learning and memory ability that bears have is also tied not only to the habitat features across the landscape, but their interactions with people. And it's not just road density or community presence or something like that. I think it's also tied to like an individual's experience in human encounters. And I'm just wondering if you have some thoughts on maybe some future models or some ways to integrate that idea into these models around learning and memory and habitat use.
Yeah, absolutely. Thanks for your question. We specifically didn't on purpose deal with human interactions because we know that they can be extremely complex, and that in itself is a whole different model. But there is, a lot of knowledge of how these factors affect bears on an individual basis. There's some work that has been done in Whistler on management approaches to bears depending on their experience to kind of test, whether different preventive measures or different management actions are affecting the way that bears interact with humans in these cases. I think that there's a lot of potential to include some of that in a model that deals with learning and memory. So for example, my model could be even more complex if you added a submodel that deals with all of these issues. So not only do you have a bear getting close to a road, but now you're accounting to what the bear's experience is specifically in that spot and how that might be different to its experience maybe in another road where it didn't encounter a human or where the experience was different. So that's one of the beautiful things about agent-based modeling is they can be as complex as you want to make them, with including different submodels and including more and more specific things in the model that can let you get to all of those complexities. That's not something that I'm focusing with on my model, but for sure there's the option of working with that in the future.
Thanks.
Great. Looks like we have some more questions in the chat. We have one from Mariella. It's for you, Ali. She says, regarding the model development, how many parameters or what parameters of it are Grizzly specific regarding generalizing to other species?
Thanks, Mariella. That's a really good question. The model that I built is generally pretty specific to grizzly bears. We did model for their specific rates of movement, their specific rates of consumption and utilization of resources as they move. So it is pretty specific to grizzly bears. We used GPS data to generate all these parameters. That being said, if you have enough information about the behavior, or if you have GPS data from other species, it could be pretty easily adapted to other species. So I like to think of it as like a base code in R that then you can input your own variables and generate it for whatever system you're looking for. So the one that I use should not be used for anything other than grizzlies, even grizzlies in other areas, because their movement can be very different. However, there is a huge opportunity for modifying it to adapt it to other species or other populations of bears.
Great. Our next question in the chat is from Sanzar, and it's for you, Melanie, along with a salute from Kazakhstan. So the question, there's a number of questions in here. So we'll start with the first one is, what is the percentage of error in this program? Is it necessary to capture, recapture, and immobilize individuals or mark them, tags on the ear to check the data?
Yeah, so far it depends whether we're talking about the face detection, which my presentation kind of concentrated on, or if we're talking about the full identification. At the moment, we have higher performance success with the facial detection, which we would expect because it's easier to find a face in an image than it is to identify which bear that is. So that's just basically the difference. So there are two parts to this identification pipeline: finding the face and then correctly identifying it. So we're working on both at the moment, and this presentation kind of looked at how we can take this like multi-species approach for all bears. So I'm not sure which aspects they're looking at the error for, but if we're talking about, I think they mean identification overall. So for brown bears, we're looking at about a 16% error rate, and for Andean bears, which we've done this preliminary model for, we're looking about 14%. So we're trying to get over 90% accuracy. That's kind of been our goal from the start, you know, as higher accuracy as possible, then the system's as usable as possible for researchers. So what we need is more data to keep training to try and make these models better. And so to do that, we gather, we kind of have two data sets going at the moment. We have a wild data set from some data that I've collected in Knights Inlet on the coast of BC over the past 10 years. And then we have a data set from Katmai National Park in Alaska, mainly from Brooks Falls area. So these are two sites of long-term monitoring for bears. So that's given us this like nice long-term data set of known individual bears. And that's what we need to train the systems. And then now we also have a data set of bears under human care. So by kind of bringing these two data sets together, that's kind of giving us the data that we need for training. So we haven't included any images of bears taken during capture or anything like that yet. I've always been interested to maybe try that, especially because people tend to take photos of bears during, you know, when they're being immobilized. So that would be interesting to try, but it's not something that we've included yet. So it's not necessarily to have to tranquilize and capture a bear to get the photo. We can just use photos of wild or captive individuals as well, as long as the individual is kind of recognizable and known.
Great. The next part of that question is how accurately the program displays the results of the difference in image clarity and technical characteristics of different camera models.
Well, at the moment, all of our models are only trained on images from DSLR cameras. What we're doing now is going back and retraining using camera trap images. And at the moment, we'll only be using one camera trap type because that's just what I've used for the past few years. So we're using Browning cameras, the 1080p version, to try and get as much clarity as possible. But we're definitely interested in looking further at how things like the image clarity affects the recognition accuracy. And anybody that's done camera trapping in temperate places knows that like the lens can get fogged, especially later in the season. And that definitely, if you're trying to do like manual individual ID, makes it a lot more difficult. So we can expect that the models may struggle as well with those kind of images, but we don't know yet. And that's something that we're going to look further into.
Great. Two more parts. So the next one is, how well can the program objectively distinguish between different bears, assuming that one and the same animal can pass at different distances in front of the camera?
Yeah, so again, because we've not fully done the analysis for camera trap images yet, we don't fully know. We have played around quite a bit with the setting up of cameras to try and get like what we call like an ideal facial image. And that seems to be putting cameras kind of diagonal to trails to try and get a kind of fairly close facial shot as the bear walks past. So it may mean that when people hopefully in the future use this technique, they may need to adjust their camera trapping method slightly just to make sure that they capture, you know, putting cameras nice and low so that they capture as clear a facial shot as possible as the bear walks by.
Great. And the last part of this question is it necessary to set the scale of translation for any external object in front of the camera, such as a stone, brush, tree, ruler?
Again, we don't really know yet about the effects of background or things in the foreground of the image. So yeah, I can't really address that question yet, but that's definitely something that we're looking into. And background and surrounding area does have an effect. Been research done on other species, specifically on detection of the animal. So that can be affected by the background. So you need to make sure that when you're doing this training, you're including lots of different types of habitats. It's one kind of thing that people sometimes, if you're using a model that is trained for like individual ID or species ID, and you're using like a pre-trained model from one habitat and you're trying to use it in a different habitat, you may not have the same level of accuracy. So it's kind of one real thing to be aware of. You need to make sure that the area that you're using this model in is representative of the training data which has been used. Otherwise, you could see much lower level of accuracy. So it's important to set cameras up in different areas and to have different types of background and potentially like foreground as well, to make sure that when you're actually doing the testing that you're getting the level of accuracy that you expect.
Great, thank you, Melanie. So our next question is from Clara from Italy, and she would like to ask Ali how she built the utility maps. And she asks, are the maps the output of an output of Maxent modeling, or is it something else?
Yeah, no, great question. Thanks, Clara. So the maps that we build, the utility map is basically a mosaic of a bunch of different utility maps. What we did was calculate the probability of usage from the grizzly bear population using the GPS data that we had access to. So for each different feature that we looked at, we had a different probability of use map that then got put together to generate a baseline utility map. Because we did it at 2 different scales, we considered variables at 2 different scales. So for example, for our small scale, we considered things like topographic complexity, food availability, and distance to roads. For our larger scale, we also considered topographic complexity, but in terms of linear features and anthropogenic features, we considered more the density of them within a larger area. And then of course, these maps are getting updated every time the bears move through that area. So if the bear has a positive experience and use that area appropriately, then it's going to have a higher utility in the future. But yeah, basically just probability of use for bears reflecting quality of use.
Great. Our next question is from Dave, and it's for you, Melanie. And I think you've partially answered this one already, but you might want to expand. He asked if you have had any discussions or collaborations with Katmai National Park or McNeil River Game Sanctuary in Alaska, since both areas have a large number of identified individual brown bears.
Yeah, absolutely. So we do have a data set from Katmai, from the bear monitoring program there. So, and we're looking to try and get some data from the last few years as well. I think our last set of data was from 2017 and previous to that. We don't have any images from McNeil. There may be another project ongoing with ID of individual bears at McNeil. So we're kind of looking into that as well. And it would be great to try and maybe collaborate with that group that are starting work there, because it's quite to try and collect this kind of data, you really do need kind of a long-term data set, because we need as much variation per individual as possible. And also that allows us then to see how accurately we can recognize these same individuals over time. And that's been one of our questions as well, is can we recognize the same bear, 10 years later? So definitely these areas of long-term bear monitoring are really important.
Great. Our next question is from Tabitha, and it's also for you, Melanie. I think you've answered part of this already, but there is a new portion. So she was wondering if you have considered additional face markers and measurements such as ear measurements or the nose, and if so, what did you find? And then she also asked, she noticed that you were using movies in your last example, and she's guessing that there's a benefit to that given the increased likelihood of more angles.
Yeah, definitely. 2 really good questions. So basically we only specify the eyes as facial landmarks, so that which basically means that those areas are used. So for the second part of the face detection, so that all of the eyes of each bear are in the same place for analysis. So what it doesn't mean is that we're specifying that the eyes are important. And the method that we use of machine learning called deep learning, it basically learns by itself based on experience. So it doesn't take anything that we tell it into consideration. It learns about the face based on the face that it sees from this labeled data set of individuals or of faces, depending if we're talking about face detection or face ID. So even though we point out these facial landmarks, that's really just so the system can crop the face, and it can kind of use the eyes as landmarks to move that face into kind of the next stage. But it doesn't necessarily use that for face detection. It just basically learns based on experience from what it sees in different kinds of dimensions. So that's kind of how the process learns. There is a way of using machine learning and specifying which area you want the network to look at. That isn't really a deep learning approach, it's more of a machine learning approach. But we didn't know, starting this, what aspect of the face was going to be important. So we wanted to use more of a deep learning approach where we just provided the image and then the networks decide for themselves what is important about that bear's face to recognize it or to detect it. So it may use the nose, it may use the eyes. We think the eyes and the nose are the most important part. So we just, make sure that those are in all of the images, but we don't fully understand. That's kind of the interesting thing about these models, that we don't really know what aspect of the face that they're using. But as long as they get the answer correct, then that's the important part. And then the second part of the question was about using video. We are going to be using video for the camera trapping part of the analysis. Sorry, I forgot the name of the person that asked the question. Tabitha, sorry. As Tabitha pointed out, there has been some research done on chimpanzees that found that using video and kind of using stills of a video and combining and basically identifying or trying to identify the animal in each still and then combining that together using this kind of like face tracking approach increased the accuracy quite a bit for chimpanzees. So we're kind of hoping that that may be the same for bears. Because, if you see one image, you may, have a fairly good chance of getting it correct, whether that's a certain bear. But if you've got 10 different images from slightly different facial poses, then hopefully that would increase the chance that you'd be able to accurately identify. I know if somebody's asking me, which bear is this? If I only see one photo, it can be quite difficult. But if they show me a few photos, it makes it a little bit easier. And so these automated networks learn in a similar kind of way.
Great. The next question is also for you, Melanie, from Oddbuyer. The question is wondering about how accessible the technique or code is to use in other populations of brown bears, if they're interested in using it, for instance, for comparing with genotyping results of individuals that are identified.
Yeah, so at the moment, we have what's called like a closed set approach. And that means that we have to train the network to recognize this certain individual. So it's kind of in this database of individuals. And then at the end, if we're giving an example image, the model, the overall BearID models will always try and see if it's one of those individuals that it's already seen. So that's what's called a closed set approach. We are looking towards what's called open set, which would give us the ability to identify and recognize new individuals that the networks haven't seen before. But it's much more difficult to do that. And it's only just being done for other wildlife. So a lot of what we do with BearID is based on what's being done for other primates. At the moment, there always seems to be a human, it's a human in the loop. So a human at that point that has to distinguish whether that is a new individual or not. But we want to try and get beyond that if we can, so that it's all automated. And then we can use it in things, like doing things like population estimates and mark recapture things. But it's just that I think we're just on the cusp of those kind of developments happening for wildlife. So we're kind of working with a few collaborators at the moment to see how best we can do that. So anyway, hopefully when that has happened, that would mean that that BearID could be used by researchers for any population of grizzlies in any area. And that's kind of our main aim, is not just to develop BearID for these specific populations, but to have it so people can use it regardless of what population they're working with. But it just takes a little bit longer to get there yet. Was there a second part to the question? I can't.
I believe that was it.
Question? Okay.
Great. Our next question is from Dina, and it is for you, Ali, asking if you could speak to why you modeled utility value the way that you did instead of using results from an RSF or SSF.
Yeah, totally. Thanks for the question. So I think the bottom line here was we were trying to focus more on the modeling aspect that modeled the learning and memory aspect of it, and really trying to understand how those two factors played a role in the emergence of home ranges. There is the potential that the results could have been more focused to the population using RSFs or SSFs, but at this point it was more like computational limitation based on the timeline on the project and the goals of the project, that we decided to just calculate it using the probability of use from GPS data.
Great, thanks, Ali. Our next question is also for you from Mihai. The question is, do you think your model can be adapted to predict movements, for example, the movements of problem bears around settlement?
Yeah, so one of the beauties of agent-based modeling is that if you get the model right, it can be used for predictive analysis. So if you get the procedure that a bear is using to move and the effects that one decision might have on future decisions, then you can for sure use this as a predictive movement. So one of the applications that we're trying to use this model for is to understand translocated bear movement and to understand how the previous experiences that a translocated bear had in one area translate to its behavior in a new area. So that is in itself a predictive use for the model. And especially, different, I mean, you can predict the reaction to a bear if you understand the importance of the previous decisions, because it is a simulation model. So you can predict the movement in a new environment. You can predict it if the behavioral rules change. So it does have that capability for sure.
Great, thank you. Our next question is from Melanie, and it is from Sandeep. The question is, does this algorithm only work for brown bear face ID, or is it useful for any other bear species? Also, how big of a training data set do you need for this algorithm to work accurately?
Yeah, so we started off focusing on brown bears, and they continue to be kind of one of our focal species. For the work that was included in the presentation, we've started to expand that, and we use Andean bears to kind of prototype this multi-species face detector and take it through to a full identification pipeline. But our sample size is quite low for Andean bears to start with, and so the data set that we have for Andean bears at the moment is from bears under human care. So we're looking at the potential of maybe supplementing that with some wild images as well, which would help us to increase that data set. So we've been kind of developing mainly for brown bears to start with, because that is the species we have the largest data set for. But we are now looking to expand to a few other species as well. But it really depends on what we can gather the training data set for these different species. So looking at whether we can gather I mean, the second part of the question was about sample size. So it's kind of an unknown, as it kind of always is with sample sizes, but we want to try and include at least kind of 50 individuals per species, and at least 20 images of each individual. And then of those 20 images, ideally they would all be taken on different days. Ideally, you know, if the species goes through like a rapid weight increase like brown bears do, then we want images at different times of year as well. So it's it's a bit of complexity in terms of the actual images that are needed, and it basically just has to represent the variation that the species shows. So if there isn't much change in like the length of the coat or the weight, then it may not be as important to have images across the years. It just has to represent the change that these different species go through to try and make the systems as robust as possible. Because the one thing that we've looked at with brown bears is if we only train on images of bears in the spring, and then we try and test on images of bears in the fall, like coastal brown bears can look quite different, so that those different times of years. So the system struggles a little bit more. So we have to include images to show the system this is how different bears can look at different times of year. So we try and include more individuals the better, and the more images of those individuals the better, but it just depends on data sets that we can accrue from different researchers. Was there a second part? I can't remember.
I think that is it. Yeah. So let's see, we have another one for you, Melanie, that says, have you looked at adult bears only? I'm wondering if you have been collecting samples long enough to ID cubs later in life.
Yeah, so we've avoided cubs of the year so far, just because they're so small, and it's difficult to, especially if you've got a litter of kind of 3 cubs or so, it's difficult to track those individuals as they go from cubs of the year to yearlings. So I think the youngest bears we have in the data set are yearlings, and they are now 7-year-olds approximately. But yeah, I don't know off the top of my hand the accuracy of those images from when they were yearlings, but again, it's something that we're interested to look at. One thing that we've noted in the last kind of year or so is that the subadult to adult age seems to be quite a vital stage. And so if we only have images of a bear when it's kind of 3 years old, and then we jump to images of that same bear when it's like 7 or 8, then the system struggles. So I think that we need to be able to try and track those individuals through time and include images of them with it between those different ages. And then hopefully the system can learn how bears change with that age. So like I've said already, it's all about the training data. So if we can include all of that information in our training data, then hopefully it will be able to perform well when it's tested on bears of any age.
Great, thank you. It looks like we have another question for Abby. This one's from Mariella, asking if you could get any reliable biomarkers from hair, and if it would not work or if it would degrade too quickly.
Yeah, Mariella, you can definitely get biomarkers from hair. We measure cortisol, progesterone, testosterone, and estradiol in hair, and it's a bit before my time on the project, but they did a lot of work looking at concentration related to nutritional status and body condition, and then those reproductive hormones with captive individuals, and looking at different times, things like that. Yeah, hair is great, and it's easy to collect, and you can just store it in an envelope at room temperature. So I would always encourage everybody to collect some hair if you're working with bears, because we can get a lot of information, not to mention, getting DNA and whatnot from it as well. So yeah, it's a good matrix for sure.
Great, thanks, Abby. Next question is for Melanie, asking if this technique would apply for nighttime images of bear species in areas where they're predominantly nocturnal.
Yeah, so we're going to be including infrared images when we're doing this retraining with the camera trap images. I mean, I find it more difficult to recognize individuals at night. So I'm kind of wondering whether the system may struggle a little bit as well. However, if we include training images of, in black and white, then hopefully it will learn to understand the differences in these types of images. The other thing that we can do as well is we can kind of manipulate images. This is done quite a bit in machine learning, particularly computer vision literature. So things like you can jitter an image a little bit to make it slightly different. And that could increase like your data set and just increase the variation of that image. So we can do things like we can change the coloration in daytime images of bears and just make them black and white and then kind of overexpose them a little bit so they represent more of a nighttime image, but we can still recognize the individual because we've seen it in the daytime. So we can do that as well as using infrared images taken from camera traps as well. So hopefully we will be looking at that. And as we're going through and labeling our camera trap data, that's something that we're picking out is whether it's an infrared image or not. And then we can compare between the performance of infrared versus daytime images.
Great, thanks, Melanie. It looks like Mariella has some follow-up questions for you, Abby.
Yes, sorry about that. So how long is that hair usable from the moment you take it? And also how long of a time frame does it show you for the...
Okay, so the first question, how long you can use it, from what I know, people have used like archive samples in museums. So as long as it's dried and stored dry in an envelope and away from sunlight, then I think you can use it for several years.
Nice.
And then the time period that it represents would be whatever the growth of that, like for bears, it would be the active season, right? You need to make sure you're not getting any new growth or after molting hair. You want to get the hair that represents like the period before, if that makes sense.
Okay, nice, thanks.
No problem.
All right, we have another question for Abby. What is the average cost per sample for protein analysis from skin? And is the presented method cheaper than microarray? Sorry for such a boring economical question. Not boring, great question.
Great question.
Yeah, great question. And we have to know these things if we think we're gonna actually implement them in our practice, right? So yeah, great question. But I'm sorry that I do not know the answer about the microarray because I'm not sure how much that costs. The protein, so I think the microarray was done in-house with our lab with Dave Jans before I got there. So I'm not sure, but for the proteomics analysis, we run it with University of Victoria, and it's about the initial development probably costs a couple thousand. Once you have your protein panel and your targets figured out, then your sample cost per sample is around like $135, I think.
Great, thank you. And there was a follow-up that that they had heard about 200 euros per sample, so for microarray. So this is cheaper.
Yeah, and actually, once you find, the upfront cost is developing it and validating it. But once you do that, like we developed this target panel, and now we've applied it to our skin samples to look at landscape variables. We've applied it to serum samples to look at reproductive biomarkers. Like kind of once you have, and that's really what this work is all about, is trying to narrow down which are actually going to be good biomarkers that are actually going to tell us things about the individuals. So once you have an idea of what that like ideal health profile would look like, I don't think it would be very expensive at all.
Great, thanks, Abby. I'm not seeing any more questions in the chat right now, so I guess I will ask a question. Sticking with the theme of the conference of conserving bears in a changing world, I'm wondering if the panel could discuss specifically like what emerging challenges your applied conservation solutions could help us overcome in the future.
Before we get into that, Lindsey, can I just address this last quick question in the chat, since just we're still on the talking about the protein work. You don't have to develop your own. We can collaborate and you can use ours. You just have to work with the University of Victoria and probably go through us to be able to use that panel and their quantitative metrics and all that kind of stuff. Oh, and good point, Aga. Yeah. Okay, go ahead, Lindsey.
Great. So yeah, open for any of the panel members for discussion, just generally how your findings might apply to our overarching theme of bears in a changing world.
I'm happy to go first. I think with my project specifically, or our project, one of the main kind of benefits that we see in this technology is to speed up the processing of data. So this kind of automated approach, maybe not just speed up, but standardize it as well at the same time. So, you know, research projects are always struggling to find funding. You know, funding is difficult to accrue, but time is as well. And so anybody that's done, you know, does camera trapping as part of their data collection knows how long it takes to process camera trap data, whether you're processing to the species level or to the individual level. And so, you know, it can take months to go through, particularly if you're working on like big multi-panel, you know, camera trapping projects, multi-stakeholder projects. And so we think that this could really help to speed up the processing of that camera trap data so that researchers can spend more time actually addressing the research questions and digging into the analysis, and less time just focusing on going through every single camera trap image and trying to work out who's in that image, how many individuals, how many, you know, what species they are. And so for species recognition, this is already kind of happening. You know, we've got websites like Wildlife Insights, which aim to do the same thing for identification of species in camera trap images. So we kind of see this as a bit of an add-on to that. You know, my ideal would be in the future that we can input a camera trap image and we can use these automated models that'll tell us, you know, how many animals are in that image, what species is it, and maybe even, you know, what age, what sex, and which individual is seen in that image as well. I mean, for me, that is kind of an ultimate goal, but I think that if we had that ability, it would allow us to, you know, ask different research questions and be able to dig into the data a lot quicker as well.
Do you think, Mel, I was going to ask you, do you think that your technique would be like complementary of a DNA hair snag for population inventory, or do you think it would eventually replace it one day?
I mean, I think it depends on the research question. I mean, if you're just looking to maybe use DNA to find individuals, perhaps this could be a cheaper and quicker method. But, you know, if you're wanting to do like things like diet analysis and you know, physiology work, then it could be complementary as well to have the paired methods. Exactly. So I think it really depends on what it is you're trying to find out, but it just offers an additional option, an additional tool to be able to recognize individuals potentially quicker than doing it via DNA.
Mm-hmm. I think it would be really helpful, because I'm thinking about how we, and this goes to your question, Lindsey, about how we monitor the population. We put up these grid cells with barbed wire to collect hair, but we always put up cameras at those locations. But if we could apply your software and be able to confirm that this was truly a unique individual, because oftentimes we'll have like 2 tufts of hair, or we won't be sure. Like I just feel like it would be a really mm-hmm. nice, complementary to that. And yeah, a lot of the physiology work that we do is trying to answer similar questions as what Melanie was talking about, and being able to tell more about the demographics of our population, and then also how they're responding to changes on the landscape, and being able to identify like kind of these hot spots or locations where, you know, maybe cortisol values are way highly extreme compared to other areas, and then be able to inform some management decisions based off that.
Mm-hmm. I think it's like expanding the toolkit, isn't it? And trying to take advantage of these new techniques that people are using. So why not, you know, try and include this in bear research as well, and try and find out as much as we can. You know, like you say, a bear is in one area at one time. Let's try and get as much information as we can from it as possible.
And just to add to that, and going back to the boring economical aspect, I mean, yes, funding is limited. So having multiple choices of how to collect information, and then, you can pair up with people that have already done camera trap work to get your genetics and your individual identification. So much potential for collaboration, and there's so much camera trap data out there that a software like this, I think, can open up the door to a lot of really new cool questions for sure.
And sharing data, sharing camera trap data is going to be really important there as well. Like you say, there's so much camera trap data that gets used to answer one question and then just sits on people's hard drives at home, mine included. And so it would be really nice to try and encourage more collaborative projects that share data, and so people can take advantage of it for different species as well.
There is I wonder if you have a similar thing. We have a hormone base, which is like a big database that you can upload your hormone concentrations for your different species if you're studying. So that, because one of our big issues in the physiology world is establishing these baselines, because I don't, if I don't have tons and tons of data, I don't really know what these changes mean relative to the baseline. Is there anything like that for your technique? Like, could people upload tons of images to help train your models?
Yeah, so there are, but at the moment they're mostly focused on species recognition. And so things like there's like Snapshot Serengeti, and there's now like Snapshot USA, even like iNaturalist information I think is available. So it's kind of, people can upload their images from the camera traps, and then it's kind of labeled in a certain way so that people can just download the kind of labeled data for that data set as well, and that can be used to help train these recognition models. But they are slightly few and far between, and like I said before, it does really depend on the habitat in which they're focused and how transferable that is to people working in different areas and different systems. So yeah, I think that's going to be really important going forward. It's something that I was kind of looking into trying to maybe get started, like some kind of online platform where people could upload their labeled data, because after we presented it the previous IBA conference, we had a lot of people reaching out to us offering their data, and so we want to try and maybe see if there's some way of kind of gathering that together and putting it online in like an open source format so people can come and use it to develop these models. Because there are people across the world looking more towards conservation technology and computer vision and focusing on wildlife conservation for that. So I think it would be great to have like a repository almost. I think there are a couple of things around, but I don't believe there's any yet that's like focused on for wildlife conservation, and not for individual ID. It's mostly for species ID. But I think it could also be interesting for sex recognition and age class recognition as well.
Great. Well, we have about 1 minute left, so I guess I will just open it up to see if our speakers have any last minute thoughts they'd like to share with us before we end.
Just want to thank the organizing committee and everybody for showing up, and the speakers. Is, I think, a great conversation, and I know we're all pretty open to continuing to talk about this. So at least for myself, if anyone has any other comments or questions, feel free to reach out. Yeah, keep talking about it.
Yeah, of course. Yeah, thanks you guys. That was great.
Yeah, thanks for so many great questions. It was great to fill the time with questions for sure.
Yeah, it was.
Great. Well, thank you again to everyone for tuning in and for the great discussion. It was great to hear from all of our speakers, and we're looking forward to the rest of the sessions.
Thank you, everyone. Thanks for joining us.